move to the first slide. Durai, I'm not sure if you need to enable something because my slides are not moving.
Oh, okay.
Thank you very much. Thank you.
Yeah.
So recently, over the last 5 years or so, we've been successful in having one of these compounds actually commercialized with an American company called Cthulhu Ventures. And this has resulted in two spin-off companies called Oncogel Therapeutics LLC, which is the American company, and Oncogel Therapeutics Pty Limited. So these two companies were founded to actually take our drug into clinical trials. And this has occurred. The drug which went into clinical trials is called DPC. The structure of DPC is presented here. It's a small molecular weight compound, quite lipophilic, quite membrane permeable. And so this particular drug actually went into an open label dose finding and pharmacokinetic phase 1 clinical trial in advanced cancer patients. So these were patients which had very, very advanced cancers which have been treated over and over with a variety of different types of anti-cancer agents. So the particular trial actually looked at DPC given orally. DPC is an interesting compound. It can be actually delivered by the oral route in the form of a tablet, but it can also be injected intravenously. And what the investigators were able to demonstrate was that the compound showed very good absorption from the gut, which is actually important, of course, for giving a compound as a tablet, and appropriate pharmacokinetics. So the compound had a half-life in humans of around about 18 hours, which is actually quite appropriate and quite good. And this was pretty similar to what we actually found in the rats. In the rats, we found the compound had a half-life of around about 10 hours. So these are very interesting compounds, and I'd like to tell you the story of actually how we developed them and why we developed them. So basically, these compounds have a very different mechanism of action. We were after a compound which showed a very different effect, a very different mechanism of actually killing cancer cells. And so we were interested in designing compounds which actually could bind iron and copper. Now, iron and copper are really very, very important nutrients which cancer cells require for growth and division. And you may say, well, okay, that's interesting, but you know, how do you actually get selectivity against the normal cells? Well, cancer cells, they take up far more iron and far more copper than normal cells because they're more rapidly growing. And in fact, if you look at the cancer cells in sections and compare them to the normal cells, what you find is that the cancer cells express very, very high levels of molecules such as the transferrin receptor, which takes up iron into the cell, but also copper transporters. The cancer cells require a lot of iron, a lot of copper for very basic processes. And one of these very important processes is DNA synthesis. DNA synthesis is essential for the replication of the cancer cells. And there's a very important enzyme, it's an iron-containing enzyme called ribonucleotide reductase, which catalyzes the rate-limiting step in DNA synthesis. So if you can target this step and prevent the generation of DNA, you're going a long way to killing the cancer cell. And indeed, these compounds induce apoptosis and cell death by binding iron. So they're not cytostatic, they're actually killing the cancer cells. Now, another very important aspect in terms of the way these compounds act is their ability to actually upregulate a very important molecule, which I'll talk about in some degree today, called NDRG1. NDRG1 is a metastasis suppressor. It actually blocks the spread of the cancer cells. And this is really very important because the spread of the cancer cells is what kills most patients. And this is something very, very unique about these compounds, that they can actually specifically block metastasis. In fact, this is the only compound that can actually upregulate NDRG1 that's known, this very, very important molecule. So it's a very different way of actually killing cancer cells. So I thought I'd just show you a couple of the structures of these compounds. This was our first lead compound. I'll talk about this compound quite a bit as we go through the talk. It's called DPC-11. 4,4-meth-T. It's a small molecular weight molecule. As I mentioned, these compounds bind iron, they bind copper, and they have 3 teeth to do this. So they have this pyridyl nitrogen here, this imine nitrogen, and this thiocarbonyl sulfur. So these 3 teeth are used to bind the iron and the copper, which are essential for growth and proliferation. Now, these particular compounds are interesting because they do something quite unique. Not only do they bind the iron and the copper, which are needed for growth of the cancer cells, but once they've actually come into the cancer cells and once they've actually bound the iron and the copper within the cancer cells, they then redox cycle to generate free radical species. And this then kills the cancer cell. And we call this the double punch mechanism. The first punch is binding the iron and the copper, which is needed for growth. The second punch is this generation of free radicals. And I'll show you a picture, a schematic of the double punch in just a second. But I'd just like to let you know this is work which goes back to around about 2004. So what I'm going to be telling you about today is compounds which have been developed over a very, very long period of time. This is not something which we've come up with in the last couple of weeks or the last couple of months. This is something which has been developing over a very, very long period of time. The compounds demonstrated very, very good activity in vitro in cell culture. They were killing the cancer cells very effectively at very low concentrations, 0.06 of a micromolar, relative to when we looked at normal cells, a whole variety of different normal cells, not just fibroblasts. When we looked at the fibroblasts, it took up to 25 micromolar of the compound to demonstrate anti-proliferative activity. So we're seeing a very, very big difference here. You only need very low amounts of the compound to actually kill the cancer cells, whereas the normal cells, it takes a heck of a lot more compound to actually inhibit the growth. So we've got selectivity, which is really important and something that you want in terms of designing an anti-cancer agent. Now, this is just a schematic of this double punch mechanism. Here we have the compound. Here it's called the DP44-meth-T. As I've mentioned before, it's very lipophilic. It satisfies all of the properties of Lipinski. So it can actually come into cancer cells very, very quickly, where it binds the iron or the copper within the cancer cells and forms a complex. And so what you can see, this is an iron complex with one chelator wrapped around the iron over this side, and then another compound, another ligand wrapped around the iron on the other side. And so what you get is a complete coordination shell. Now, this is the first step in the double punch, the binding of the iron or the copper, which prevents the cells from using the iron and the copper for growth. Then what you get is these compounds then generate free radical species. So they actually, what happens is the binding sites create an appropriate microenvironment, an appropriate redox potential to be reduced by intracellular reductants such as NADH or FADH, and you get the generation of iron 2 or copper 1 complexes, and then they interact with oxygen to generate the free radicals, which then go on to kill the cancer cells. So this is a very unique way of actually killing the cancer cells. Now, one of the aspects which I already discussed very briefly is the ability of these compounds to actually overcome this triad of death. And this is just a slide just demonstrating the triad of death here, composed of these 3 axes: tumor growth, primary tumor growth, metastasis, spread of the cancer, and drug resistance. So in order to actually effectively inhibit the growth of cancer, you need to go after each of these axes. And the very interesting aspect of these compounds is that they actually have multiple molecular targets, which enables this to occur. So in terms of inhibiting metastasis, these compounds upregulate this protein NDRG1, this metastasis suppressor, which blocks the spread of the cancer cells. And I'll show you some data in just the next couple of slides, just demonstrating how effective they are at blocking the spread. Then these compounds also affect other molecular targets in the cell to block primary tumor growth. So for instance, they can upregulate tumor suppressors such as P53 to actually block cell cycle progression and a variety of other processes. And finally, another aspect which I'm going to talk about today in quite a lot of detail is the ability of these compounds to work by a very unique mechanism to overcome P-glycoprotein-mediated drug resistance by actually being transported by a pump called P-glycoprotein, which is a very important molecule which actually imparts resistance on the cancer cells. And again, resistance is really important in terms of the pathogenesis of the cancer. So how good are these compounds? Well, this is some studies in vitro using nude mice. And so what we've done here, we've taken these animals and we've injected them subcutaneously with a tumor. We've allowed the tumor cells to actually grow and divide, which occurs over about a week or two. And we allow that tumor to grow to around about 100 millimeters cubed. And then once we have the primary tumor, we then begin tail vein injections to actually test the systemic effect of these compounds on the growth of the primary tumor in the mice. And this is actually a melanoma. This was one of the first tumors which we were looking at. We chose melanoma because we were interested in trying to develop a compound for highly resistant solid tumors. We weren't interested in tumors which were very susceptible. We were after the tumors which don't respond very well to current chemotherapies. And so melanoma was a good choice. And what you can see is here, some studies, after 5 to 6 weeks of treatment, you can see that our first compound, our first lead compound called DP4-FT, has very markedly decreased the growth of these melanoma tumors. So this was really very exciting in terms of their activity. We then went on to look at a whole variety of other types of tumor xenografts in the mice. And this was basically important because we were after a compound which would be acting like something like a penicillin, having an effect on a whole variety of different kinds of cancers. And indeed, this is some studies looking at lung cancer. Again, it's using the in vivo nude mouse model. And so this is a lung carcinoma. This is the control tumors. They grow very well in the mice. But after treatment with DP4-FT, and this is intravenous therapy, by the way, after only 12 to 14 days, you can see the very marked inhibition of growth induced by this compound. We then looked at ovarian carcinoma cells, another very common tumor. Again, we see a nice inhibition of growth. This is a neuroepithelioma. So again, the compound was showing good activity at blocking growth over very short periods of treatment, over 10 to 12 days of treatment. And again, this is the melanoma. So this was very encouraging studies for us very early on in the process in terms of trying to develop these compounds. And so we went on to do far more extensive studies. This is another tumor. This is looking at pancreatic cancer. Again, we looked at a very aggressive tumor. This has about a 95% death rate. And so a very, very difficult to treat cancer. And this is studies done over a much longer period of time, up to 42 days. You can see the growth of the tumor is very, very quick and very avid in these animals. So we get a very large tumor quite quickly. You can see that we've also here assessed our drugs relative to a standard chemotherapy, which is called gemcitabine. Gemcitabine has quite a good effect at blocking the growth of the tumor. It had about the same effect as our first lead compound, our first generation compound called DP4-FT. So DP4-FT and gemcitabine, pretty similar activity. However, the important thing here was a second generation compound, which was called DPC. DPC totally blocked the growth of the tumor. So this was very exciting for us. You can see the very marked effect of DPC here relative to the control and relative to the standard chemotherapy gemcitabine. Now, another very important aspect was that the compound inhibited the growth of the tumor without having a marked effect on the weight of the animal. Now, this is important. If you give an animal an anti-cancer drug and it begins to lose weight, you know that the compound isn't well tolerated. But in this case, the compound was well tolerated over this period, quite a long period of treatment, of intensive treatment, 5 days a week for up to 42 days. So again, very, very good news. We then went on to look at DPC given orally via gavage to these animals. And so what we can see here is a study again in the nude mice, but this time the drug was administered via the mouth of the animal via gavage. Again, you can see this particular tumor, which is a lung cancer, grows very nicely if you just give the vehicle control. But when you administer DPC totally blocked the growth of the tumor, which was really great, without any effect on the body weight of the actual animal, which again demonstrates that it was tolerable, and no effect on any of the major organs that you can see from the histology. And this is just some photographs of the heart. So the compounds were very tolerable, and this was also a very exciting result for us. We then looked a little bit more at the toxicology of these compounds and the tolerability just by assessing the effects of the weights of the various organs compared to the tumor. And out of all of these organs which we've examined here over this period of treatment via the oral administration, you can see that the only tissue that's losing weight here is the tumor, which is great. That's exactly what we want. We want to inhibit the growth of the tumor, and we don't want to have any effect on the normal organs. So again, selectivity. The compound was showing selectivity. Now, another aspect, which is really quite critical, is the fact that these compounds are chelators. They bind iron, they bind copper. And so we were somewhat concerned that we would see hematological effects, that is, effects on the blood, because obviously red blood cells need a heck of a lot of iron to generate hemoglobin. So we thought to ourselves, well, it's very important to assess the effect of these compounds on the normal hematology of these animals. And what we found was that, to our great delight, there was absolutely no effect on the red blood cells or actually any of the hematological parameters over the period of treatment at the doses which we were giving. So this was quite important. And in fact, it was also validated when we actually went to the human clinical trials. We also didn't see any effect on the normal hematology in the humans. And it's because of the very low dose which you need to actually treat the patients. You only need 10 milligram per kilogram in the mice or 20 milligram per kilogram in the mice, which is really very, very low. And at these doses, you don't get any effects on the normal metabolism of metals, which is really good. So you don't affect the normal hematology. So up to this point, I've shown you the effect of the compounds basically on primary tumor growths on xenografts using a lot of different adult cancers. We then thought, well, it's also important to look at childhood tumors such as neuroblastoma. Now, neuroblastoma is a very, very aggressive childhood cancer. And again, we thought this was a really important tumor to actually demonstrate the activity of our compounds. And so again, what we did here, we've actually done a study looking at neuroblastoma tumors in the adrenal glands, implanted into the adrenal glands of these animals. And you can see that the compound DPC, our second generation compound, has actually resulted in a good decrease in the growth of the tumors as well. So we were getting very broad activity of these compounds in a whole variety of different kinds of tumors. And we think that this is probably due to the fact that these compounds are binding iron and copper. Iron and copper are needed for so many different essential processes in the cells. So these compounds have multiple molecular targets. And that's important because cancer is a moving target itself. It keeps changing as it develops. It'll change, it'll alter its molecular target. So it's important for a drug to actually have a variety of molecular targets so that you can get a good anti-tumor response. So basically what we can see here is that these compounds come into the cells, they bind the iron, and they have a number of different effects. So for instance, they upregulate the protein P21. P21 is a cyclin-dependent kinase inhibitor. It blocks the cell cycle. So this is, again, a very good response. There's also a decrease in another protein, which is called cyclin D. Cyclin D1, in fact, cyclin D1 is really important for the progression of the cells through the cell cycle, through the G1 phase of the cell cycle. So decreasing cyclin D1 is another very, very good indicator. I mentioned earlier about ribonucleotide reductase. This is the rate-limiting step in DNA synthesis. Again, the ability of these compounds to bind iron prevents the iron from actually being incorporated into this protein and prevents its enzymatic activity of reducing ribonucleotides into deoxyribonucleotides, shutting off DNA synthesis. Now, due to the fact that you're shutting off DNA synthesis, this results in a response which results in upregulation of a tumor suppressor protein, which is called P53. It's one of what they call the guardian of the genome. And this is a transcription factor which actually can very markedly shut down the cell. And so this upregulation is also very, very important. And finally, we have here this NDRG1, which is something that my laboratory has basically focused on the last 15 years or more. And we've been able to characterize that this molecule, this NDRG1, is a very effective metastasis suppressor through its ability to actually block a whole variety of different cellular signaling pathways, which actually shut down the ability of the tumor to spread. So what you get basically as a subject of this barrage of anti-cancer activity is a whole variety of responses. So you get a G1S arrest in the cell cycle, you get inhibition of proliferation. Now this is just not cytostasis, this is actually cytotoxic. So you get apoptosis and you get cell death. And not only that, you also get metastasis suppression, which is really very important. Metastasis plays a very, very big role in killing many different patients. So it's important that these compounds can block the metastatic process. And this is just, I thought I'd show you this slide because I think it's very interesting. Our work was published, of course, and then subsequently it was independently validated, not once, not twice, but by many different independent investigators throughout the world. And so this is one particular case of independent validation of the activity of our compounds, demonstrating how effective they are at blocking the spread of the cancer. So this is on day 0. This is studies done taking NOD-SCID mice and injecting them into the heart with breast cancer cells which have the propensity to migrate to bone. Now, that's a very nasty aspect of breast cancer pathology, that these cells actually metastasize and then spread to bone and cause all kinds of problems. So what they've done here, they've taken these nasty cancer cells, they've injected them. These cancer cells have a bioluminescent label, so you can see the cells. Once they've injected them into the heart, the cells are pumped around the body by the heart, and they're spreading throughout the body of the animal. So this is day 0. This is the saline control at the top. And this is after 9 days and 3 injections of saline. And what you can see is that the tumors have become localized here, probably into the brains of the, into the bones of the skull, probably here to the rib cage, perhaps, and to the vertebral column, perhaps here to the pelvis, and again here to the head. So the cancer cells have spread very, very effectively over 9 days. This is the control at the top. We then look at the effect of our compound, our lead compound. And what you can see here is the studies with the animals after the injection of DP4-FT. You can see that the DP4-FT, after only 9 days and 3 injections, has almost totally blocked the growth and the spread of the cancers. Compared to the control at the top, you can see just very, very minor tumor growths here. So DP4-FT, our lead compound, has been demonstrated here by a totally independent group in the US to very markedly inhibit the growth of the cancer cells, which was in very, very good agreement with our studies. So one of the things is, when you try and develop a new anti-cancer agent, is that you want to try and understand how it actually works in the cancer cells. And so we did this, we characterized these compounds and their ability to actually affect molecular targets and also to affect various organelles within the actual cells. And what I'd like to tell you about is this series of studies demonstrating that our drugs love lysosomes. Lysosomes are the organelles in the cells that actually digest proteins and organelles and actually break down proteins and then liberate the nutrients. So it's a very, very important process in the cancer cells, in all cells. The lysosome is known as the garbage man of the cancer cell, or the normal cell. It cleans up the cell and it helps the cell. Now, the fact that these compounds are actually, getting into the lysosome, which is what I'm going to show you today, is actually very, very important in terms of the ability of these compounds to actually overcome drug resistance, which is a huge problem in the clinics where the cancer cells become resistant due to the expression of transporters such as P-glycoprotein, where no longer the standard chemotherapy can kill the cancer cells. So they're resistant to standard chemotherapy. So the fact that these compounds are entering the lysosome, targeting the lysosome, is actually really important to understand in terms of understanding how these compounds are working. So I'd like to tell you a little bit about these studies. So the first thing that we were interested in is looking at where these compounds actually go within the cells and how they actually bind the metals. And to bind metals in cells, you can very sensitively assess this using a number of different radioisotopes. And classically, we use the isotope iron 59 to actually look at the ability of these compounds to bind iron, but also another isotope, which is called copper 64, to look at their ability to bind copper. And to our surprise, when we looked at the ability of our compounds to actually bind the copper in the cancer cells, what we found was that once they bind the copper, they seem to prevent the copper from being released from the cancer cells. And in fact, the copper accumulates with the compound. And so this was quite a strange observation, that once the compounds come into the cell, they bind the copper and they seem to get stuck in the cell and they seem to accumulate in the cell. And so we started to think, well, what on earth is happening here? Where are the compounds actually getting stuck? Where are they actually accumulating? And so we did a cellular fractionation of where all the copper was after the treatment with our compounds. And what we found, to our surprise, was a lot of the copper binding compound was actually found within the lysosomal fraction. And so this was quite a surprise at the time, because we didn't have no idea that they'd be found in the actual lysosomes. But the lysosomes, as I mentioned previously, they're really very important for the cell. And so they could be a very important molecular target. So we started to think about, well, how are these compounds actually getting stuck in the lysosome? Now, one of the aspects of biology is that you'll probably understand that the chemistry of the compound will dictate the way it actually works in the cells. So the chemistry of the compound dictates its pharmacological effects. And so we started to think about, well, what are the chemical properties of these compounds that lead to them being accumulated within the actual lysosomal compartment? So this is a study looking at potentiometric titrations, looking at the charge of the compound as a function of pH. Now that's important, because if a compound is neutral at the pH of the blood, 7.4, then the compound will be able to get into the cancer cells very, very effectively. And indeed, when we looked at our compound and looked at its ionization at pH 7.4, which is the pH of the blood or the cytosol within the cells, we found that the compound was totally neutral. It didn't have any charge. So this meant that the compound could get through the membrane very, very nicely at the pH of the blood. Now, the fact was, in the previous slide, I showed you that the compounds were accumulating in the lysosome. The interesting thing about the lysosome is that it's at pH 5, which is quite a difference to the pH of the blood. When we looked at the pH 5 and then looked at the charge on our compound, we found around about 16% of the compound actually had a charge on it at pH 5. So we started to think about this, and if you think about it, once the compound has come through the cell, once it's in the blood, it's at pH 7.4, it's neutral, it'll come through the plasma membrane very, very easily because it is neutral. It'll come into the cytosol, which is also at around pH 7.4. But the moment that it goes into the lysosome, it's going to get a charge on it. It's going to get this charge, which will block the compound from being able to diffuse back out of the lysosome. So basically, it'll come in, get a charge, and then it'll begin to accumulate within the lysosome. So we thought, well, perhaps this explains how these compounds are actually getting stuck in the lysosomes due to their charge. So we started to think, okay, well, we're getting this accumulation of the compound within the lysosome. Is it actually affecting the lysosome just because the lysosome is such a critical organelle? And so we looked at this using some microscopy, looking at live cell imaging and taking cancer cells. These are the cancer cells here, as you can see. The cancer cells have been labeled in this case with a dye which is called acridine orange. Acridine orange accumulates within the lysosomes, and basically you can see all of the beautiful lysosomes being labeled here in the control cells. So what we have is lysosomes. All of these little baby orange lysosomes are all very, very beautifully shown here in this particular picture. The interesting thing was when we take these cells and then we incubate them with our compound, which accumulates within the lysosome, what we actually found was that the compound targeted the lysosomes and induced these lysosomes to burst. And in fact, you can see quite a large reduction in the number of lysosomes here relative to the control. And the other thing which was happening was that you were getting bursting of the lysosomes, and you could see that actually down the microscope. You could actually see these little lysosomes popping in front of your eyes in the presence of these compounds. So these particular compounds were very active at damaging the lysosome. So we started to think, okay, well, that was with the acridine orange. What about other markers of the lysosome? We thought we'd better check other markers of the lysosome to be absolutely 100% careful. And so we used a couple of different, very specific markers of the lysosome. The lysotracker red, which stains all of the lysosomes here, this beautiful red. And another one, which is called pepstatin A BobDippy FL conjugate, which also accumulates within the lysosomes. And again, you can see them all here, very beautifully labeled in green. Now, this is using a special microscope, which is called a confocal microscope, which enables you to see if these labels are in the same place. That is, if the lysotracker red is in the lysosome and the pepstatin A is in the lysosome. If they're in the same place, the red and the green will merge to form yellow. It's just like mixing paints. When you mix red and green, you get yellow. And so what it demonstrates is that these two dyes are in exactly the same place. They're in the lysosome. So this is the control. You can see all the lysosomes beautifully labeled here. You then take these cells and you incubate them with this compound that was bursting the lysosomes. You can see quite clearly that we've lost a lot of these lysosomes after treatment with the compound. So all of this granularity, which is the lysosomes, has disappeared. Same thing goes here with the green stain. And you can note with this particular stain is that not only have we lost the granularity, but we can see the actual dye has come out of the lysosomes and it's spread throughout the cytosol of the cell. So what's happened here is the compound seemingly has damaged the lysosomes, and the dye is now leaching out of the lysosome and coming into the cytosol. Now, that is a catastrophic event. That damage to the lysosome, followed by the leakage of enzymes, proteolytic enzymes, out of the lysosome into the cytosol, can actually induce apoptosis, which is the cell death, the cancer cell death. And you can actually look at that by examining the cleavage of BID, a protein called BID, which is a pro-apoptotic mediator of the apoptosis. And what you can see here, when we look at the cells which have been treated with our compound, you can see an increase in the cleavage of BID, which is very, very marked in these particular cells. So this was really very interesting. We were getting induction of apoptosis after the damage of the lysosomal membrane. So this was quite interesting. So the next question was, well, okay, we've got the compound accumulating in the lysosome, we've got the compound damaging the lysosome, but how is it actually doing that? Now, you'll remember that these compounds have this mechanism of being able to bind metals and become redox active, particularly iron and copper. And so we started to think, well, perhaps these compounds are coming into the lysosome, binding copper and iron in the lysosome, and then generating free radicals to actually kill the cancer cell. And so what we can see here is a study done to look at whether these compounds were actually inducing the generation of free radicals. This is in the test tube. And so we're looking here at taking the compounds, adding them to a test tube, and then assessing what we call DCF fluorescence. DCF is a compound which becomes fluorescent after it interacts with free radical species. And so what we've done here, we've taken the compound, we've added it to the DCF, and then we've added various metals to the compound. When we added the copper to the compound, there was a huge flux of radicals, of free radicals. And so we got a huge increase in DCF fluorescence. And this was really very, very marked after copper, and far, far less than what we see after iron. So this seemed to be a copper binding effect that we were actually witnessing here. So we started to think, well, maybe it's the ability to bind the copper in the lysosomes to actually result then in the generation of free radicals which are damaging the lysosome. So we started to think, well, let's have a look at this to see if this is true. And so what we did, we took the cells and the tumor cells, and we incubated them with various concentrations of the compound in the presence and absence of agents which can actually remove free radical species. So one of these agents is an antioxidant, which is called N-acetylcysteine. N-acetylcysteine actually mops up free radicals. It prevents free radical damage. And so we were starting to think, well, if we can mop up those radicals, perhaps we can prevent this lysosomal damage. And indeed, when we did these studies, we took the cells, we incubated them with the compound itself. The compound in the red line is very effective. It's blocking the growth of the cancer cells very, very effectively. If we take those cells and then we add to them the N-acetylcysteine to increase the levels of glutathione, which mop up the radicals, what we found is that the compounds became far less cytotoxic. So if you prevent the ability of the compound to generate radicals, you prevented the anti-proliferative activity quite nicely. If you did the opposite, instead of taking away the free radicals, if you could actually enhance the free radicals, what actually you can do, you can take these cells and you can incubate them with a substance called BSO. Now, BSO acts totally opposite to N-acetylcysteine. N-acetylcysteine increases the levels of glutathione, this antioxidant in the cells. In contrast, BSO actually inhibits glutathione generation. Now, if you inhibit glutathione generation and you then add this to the cells, what actually happens is that our compound becomes far more effective. It can kill these cancer cells far better because now there is no antioxidants to mop up these free radicals. And so our compound can damage the lysosomal membrane far more. We then looked at that using the microscope again and taking the cells and labeling them again with acridine orange to label up all of the beautiful little lysosomes, which you can see all here in orange. We then can take our cells, add the copper compound, the copper DP4-FT, and you can see that the copper complex destroys all of the lysosomes very, very quickly and very, very avidly. But if you take these cells and you add the N-acetylcysteine, the agent which blocks, which actually increases the levels of the glutathione in the cells, if you increase the glutathione, you mop up all of the radicals, you prevent the generation of the radicals, you prevent the lysosomal damage, and you save the lysosomes. So by increasing the levels of glutathione, you can prevent the lysosomal damage, demonstrating that the free radical generation is really very, very important in terms of the way these compounds are killing the cancer cells and damaging the lysosomes. So what we have here is a very unique way these compounds work to actually kill cancer cells. They are neutral at pH 7.4, as I told you, which is important for getting into the cancer cells. They can come into the lysosome, and the moment they hit the lysosomal pH, what actually happens is they gain a charge. When they get a charge, they can no longer get out of the lysosome, and so they accumulate within the actual lysosome. Now, as I mentioned, the lysosome is a really very, very important place in the cell. It recycles proteins, iron-containing proteins, copper-containing proteins, all proteins. When this happens is the proteins are broken down into amino acids. They can then release their bound iron and their bound copper. these compounds, they're in the lysosome, they're ready, they bind the copper, and they can begin to redox cycle to generate free radicals. And we could see this in the test tube. We could see that when these compounds bind the copper, they actively generate these free radical species, which are highly toxic, which permeabilize the lysosomal membrane. And we could see in the confocal microscopy, I showed you the confocal microscopy images demonstrating the release of the cathepsins, these proteolytic enzymes, out of the lysosome and actually into the cytosol. Now, those cathepsins are very, very important because they're pro-apoptotic and they cleave this molecule BID. I showed you the Western, the BID is cleaved, and then you get this pro-apoptotic BID. BID generates the classical mitochondrial apoptosis, which leads to the release of cytochrome C and cellular death. So this is cytotoxic, as I mentioned before. It's not cytostatic. So we're getting a very, very interesting, very unique way of actually killing cancer cells through the activity of these compounds. And in fact, the fact that these compounds love the lysosome and kill the lysosome was actually very important for understanding how these compounds overcome drug resistance, P-glycoprotein mediated drug resistance. Now, drug resistance in cancer is a huge problem in the clinics because what happens is patients are given drugs, standard drugs. The drugs work for 6 months or so, but then after that, they gain resistance by expressing molecules such as P-glycoprotein. P-glycoprotein is a pump, and it pumps drugs out because it's expressed here on the plasma membrane. So drugs such as doxorubicin, DOX, or vinblastine. When doxorubicin comes into the cell, it actually goes to the nucleus where it actually intercalates into DNA and results in inhibition of topoisomerase. Now, in the presence of cells that have the P-glycoprotein, the DOX is basically thrown out of the cell, it's pumped out of the cell, and it prevents the compound from killing the cancer cells. So this is really very, very important. This is a very important mechanism by the way cancer cells can actually become resistant to cytotoxic drugs. So we were very interested to see if our drugs, which work by this very unique mechanism, could overcome P-glycoprotein resistance. And so we did some studies looking at this using cells which had become resistant to vinblastine. So these are what we call resistant cells. They have very high levels of the P-glycoprotein pump. So they're very active at being able to pump out these cytotoxic drugs. And so when you add a drug like vinblastine, this cytotoxic agent, it takes an incredible amount of drug to actually kill the cancer cells because the P-glycoprotein just keeps on pumping out the drug. It prevents the toxicity of the cytotoxic drug. So this is the standard picture, what you find in many different kinds of cancers, this resistance profile, which prevents the action of the anti-cancer drug. So in contrast to the standard anti-cancer drug, we thought, let's have a look in these same cells at the effect of our compound to see if there was any difference. And what we found was really quite surprising. When we looked at these cells which had high levels of P-glycoprotein, these cells, these very nasty tumor cells which can't be killed by the vinblastine, were actually being killed by our drug. In fact, our drug was far more effective in the P-glycoprotein expressing cells than in the cells that didn't have any P-glycoprotein. So this was really interesting. Somehow, our drug was killing these very, very resistant, very nasty tumor cells. And it appeared that our drug was working better in the presence of the P-glycoprotein. So the big question was, well, how could our drug be doing this? This is really quite amazing that you have a drug which can actually kill these very, very resistant cancer cells. And so we started to think about this, and you can look at these kinds of questions using two kinds of cells. That is, cancer cells that don't have any P-glycoprotein, which are called KB31 cells, and you compare those to cells which have huge quantities of P-glycoprotein, and they're called KBV1 cells. And then you can look at the differences in the way these cells respond. So in the cells without P-glycoprotein, if you add doxorubicin, the doxorubicin works very well. It kills the cancer cells very quickly, because what happens is the doxorubicin comes into the cancer cell, goes to the nucleus, kills the cancer cell. There's no P-glycoprotein to pump it out, so it's very, very effective at killing the cancer cells. In contrast, if we look at the cells with P-glycoprotein, what happens is you add the doxorubicin, and it takes an incredible amount of doxorubicin to kill these cells. Because what happens is doxorubicin comes into the cancer cell, but immediately it's pumped out again. So it never has a chance to kill the cancer cell. So this is a big problem with this standard anti-cancer therapy. We can do a little bit of a trick here. We can add a P-glycoprotein inhibitor, which is called ilacrodar. Ilacrodar blocks the pump. If you block the pump, then what happens is no longer will the doxorubicin be shot out of the cancer cell. It can now go to the nucleus, and it can kill the cancer cell very, very effectively. So this is something that happens. This has been well known for many, many years, 20 or 30 years. This is how many drugs become resistant to, many cancers become resistant to standard chemotherapy through this particular mechanism involving P-glycoprotein. Now, the big difference here is when we come and use this system, but use our drug. If we use our drug, we see a very different effect relative to the doxorubicin. When we look at our drug in the cells that don't have any P-glycoprotein, our drug isn't working terribly well in the cells that don't have P-glycoprotein. So this suggests immediately that our compound could be using P-glycoprotein somehow to kill these cancer cells. Indeed, when you look at the cells with P-glycoprotein, our drug is working really well. It's killing these cancer cells extremely well. So again, it suggests our drug is using P-glycoprotein to kill these cancer cells. You can then take these cells with P-glycoprotein and you can inhibit the P-glycoprotein using this ilacrodar to block the pump. If you block the pump, if you block the ability of this P-glycoprotein to work, we then lose the activity of our special compound. So again, this says that this compound is using P-glycoprotein to kill the cancer cell. So this was really very interesting. We were killing these very resistant cancer cells more effectively by the ability of these compounds seemingly to use P-glycoprotein. But how was this happening? That was the big question. How was it happening? So we started to think, well, maybe, okay, we know that P-glycoprotein is important for the action of our drugs. So maybe somehow P-glycoprotein is transporting our drugs. Maybe this is what it does. It transports drugs. So perhaps maybe P-glycoprotein is transporting our drug. And so we looked at this with what we call a PGP substrate assay, looking at the ability of P-glycoprotein to act as an ATPase, to actually burn ATP. And what we found was quite interesting, that the compound was actually a P-glycoprotein substrate, particularly this copper complex, which was going into the lysosomes. It was really a very, very nice substrate, being much better than standard substrates of P-glycoprotein, such as verapamil. So this was really interesting. It told us, it gave us some suggestion that P-glycoprotein could transport our drug. But it didn't help us very much, because if this is the case, that our drug is being transported by P-glycoprotein, and if the case is that P-glycoprotein is only present on the cell membrane, what would happen is our drug would come into the cell and then it would be pumped out very, very quickly, which would prevent its activity. Now, we were seeing the absolute opposite. Our drug was coming into the cell and P-glycoprotein was somehow increasing, not decreasing. It was increasing the activity of our drug. And so we thought, well, there's something else going on here which we quite don't understand. Classically, if you go to the textbooks and you look at P-glycoprotein, what you'll see in the textbooks is a cell which is illustrated like a bubble. And what you'll see is the P-glycoprotein sitting on the cell surface and the P-glycoprotein pumping out. Now, cells aren't bubbles. They're alive. They're working. They're metabolically active. They endositose their plasma membrane. Now, what happens when a P-glycoprotein is endocytosed? Well, you can see what happens. When it's on the cell surface, it's pumping out. But when you get the endocytosis, the internalization of the P-glycoprotein into the cell, you can see what happens. There's a revectorization of the pumping. So it's not pumping out now, it's actually pumping in when you get the formation of an endosome. Now, some of these endosomes can mature into lysosomes. And if this process continues, this pumping in, what we're going to get is P-glycoprotein substrates, such as our drug, being pumped into the lysosome. And as I told you before, the lysosome is one of the key targets of our drugs. Our drugs love the lysosome. So if our drugs could be pumped more effectively into the lysosome, then we would get much better anti-cancer activity. And this would explain our observation that P-glycoprotein was acting to enhance the activity of our drug and kill these very highly resistant cancer cells expressing very, very high levels of the P-glycoprotein. So we started to think, okay, can we prove that P-glycoprotein is not only present on the cell surface, but can be actually seen inside the cells and endosomes, and more particularly in the lysosomes? And so we started to look at this. We started to see where the P-glycoprotein was in the cell. And again, we went back to this technique of using the confocal microscope. And what we can see here are studies done with cells expressing high levels of P-glycoprotein, and we're looking at the expression of the P-glycoprotein in the cell. And so you can see the P-glycoprotein here under the microscope. You can see the P-glycoprotein is present beautifully on the cell surface. You can see it here, beautifully expressed on the cell membrane. But if you look very closely at this image, you can actually see these little baby dots within the cell, and they look somewhat similar to lysosomes. So we started to think, well, could this be the P-glycoprotein in lysosomes? And you can actually stain the lysosomes very nicely using a marker called LAMP2. They use an antibody to detect the LAMP2, which is a lysosomal protein. And so when we did this, and we looked at whether the P-glycoprotein was actually present within the lysosomes, it's just like mixing paints, as I said before, with confocal microscopy. Red and green gives yellow. And so what this told us was that the P-glycoprotein was not only present on the cell membrane, but it was actually present in the cell and in the lysosomes. And this was quite interesting. We then confirmed these studies, which were done using confocal microscopy, using centrifugation. And again, it demonstrated the P-glycoprotein in the lysosomes. We looked at the nucleus as well with confocal microscopy. Again, there was no colocalization with the nucleus. We also looked at mitochondria and looked at the colocalization of P-glycoprotein with mitochondria, and there was none. The big correlation between P-glycoprotein and the lysosome was really very, very marked in these cells. So this suggested that what we had was not only P-glycoprotein on the cell surface, but also P-glycoprotein present within the lysosome. Now, if you think about it, that's quite interesting, but it could also be interpreted from a point of view that, well, you know, lysosomes are the garbage men of the cells. Perhaps your observation that the P-glycoprotein in the lysosomes is just, can be explained quite simply by the fact that the lysosomes are breaking down the P-glycoprotein. And so you see it in the lysosomes being co-localized within the lysosomal fraction because they're just breaking down the P-glycoprotein. So we needed an assay to demonstrate that these pumps, these P-glycoprotein pumps, were still active and were still bringing in P-glycoprotein substrate. So that was the next step. We wanted to test whether these pumps present on the lysosome were actually active. So again, we went back to the microscope. Again, we looked at doxorubicin. Doxorubicin is this PGP substrate which can be transported by the P-glycoprotein. And we looked at cells highly expressing P-glycoprotein, and we compared them to cells which didn't have any P-glycoprotein. And so we've added to these cells the doxorubicin. Doxorubicin is a big conjugated molecule, and it's actually inherently fluorescent. You can see it down the fluorescent microscope. It's bright red. And so what we can see here are these cells beautifully labeled with the doxorubicin. When we look at the co-localization between doxorubicin and the lysosomes using the LAMP2 stain, red and green gives yellow. So what we have here is doxorubicin in the lysosome. So indeed, the doxorubicin seems to be transported into the lysosomes in the presence of P-glycoprotein. But importantly, if we block that P-glycoprotein with alacridar, prevent it from working with the inhibitor of P-glycoprotein, we see a big change in the doxorubicin. No longer is the doxorubicin out in the cytosol, out with the lysosomes. We can see a big change when we add alacridar. The doxorubicin goes to the nucleus. And so you can see the doxorubicin changes its distribution. It's now in the nucleus. So the red of the doxorubicin with the blue of the DAPI stain, it's a nuclear stain, red and blue gives purple. So what's happened here is we've blocked that P-glycoprotein transporter using the alacridar, and the doxorubicin has then gone to its natural target, the nucleus, in these cells which are hyper-expressing the P-glycoprotein. Now, if we look at cells that don't have any P-glycoprotein, the doxorubicin just goes straight to the nucleus. It doesn't even bother about the lysosome. It actually just goes straight to the nucleus where it actually intercalates into the DNA. So what we have here is a demonstration that this pump on the lysosome is actually functional. It can pump substrates into the lysosomal compartment. So we then thought, okay, well, this is interesting. We know our drug loves the lysosome. So could this be important in terms of the action of our drug? And so we decided to look at this. And so we took the cells and we labeled them up again with the lyso tracker. So you can see all of the lysosomes beautifully stained orange here with the lyso tracker. If we then add our compound is rapidly taken up into the lysosomes. It destroys the lysosomes very quick. So you can see the loss of all of the lysosomes. But if we take those cells and we add alacridar, if we block the action of that pump using alacridar, we prevent the drug from coming into the lysosome. We prevent the damage to the lysosome. So we have all happy lysosomes here after we treat with alacridar. So what this is saying is that this pump, this P-glycoprotein pump, which is responsible for resistance, is facilitating the action of our drug getting into the lysosome, where it works very nicely to bind the copper, to damage the lysosomal membrane and kill these highly resistant cancer cells. So this is in vitro. This is all in cells and culture. We wondered, can we do a clever experiment in animals, in mice, to look at whether this mechanism is actually occurring in vivo in an animal? And so what we did, we took the little nude mice again, and this time we've injected them with the tumors. So we did two tumors. The tumor with the P-glycoprotein, the KBB1 cells, was placed on the left-hand flank. The tumors without the P-glycoprotein were placed on the right-hand flank. So what you have is one little baby mouse, two tumors with and without P-glycoprotein. We allowed those tumors to grow, so you have primary tumor masses on the animal, and then we begin injection into the tail vein for 9 days. And then we looked at the growth of the tumors in these animals. And what we can see here is that if we looked at the tumors which don't have any P-glycoprotein after treatment of the animals, you can see some effect here. There is a reduction in the growth of the tumor relative to the vehicle, but it's not very marked. The big story here is that when we looked at the tumors expressing the P-glycoprotein, you can see a very nice decrease in the growth of these P-glycoprotein expressing tumors. So not only in vitro, but also in vivo, we get this effect of the P-glycoprotein actually enhancing the uptake of the drug into the lysosome. And this is where the drug likes to get to kill the cancer cells. So we have very, very resistant cancer cells now being susceptible to the action of our drug, which we think is really very, very important. So by understanding the mechanisms of how these drugs work is very important in terms of potentially also developing even better drugs to actually result in better treatment for the patients. So I'd just like to thank everyone involved with this work, which is a work which has been undergoing, still remains going in my laboratory over the last 25 years or more. And I'd just like to thank everyone involved in my laboratory, including Jurai, who was one of my